Who we're just missing? Ash. Kennedy. Cool. Okay, sounds good, Josephine. Okay. How's everybody doing today?
Good.
Good. Made it mostly through the week. It's Thursday. But yeah, let's go ahead and get started. So I think what we'll do today, because if you haven't sensed it yet, I'm a big fan of icebreakers. We're going to continue down that path. Okay. But the first thing we're going to do is a little bit of a moving poll. Okay. And you can interpret this characterization however you like. We're going to go cats versus dogs. And any way you so choose or you want to interpret that. So when I just say the words cat versus dogs, are you what I say? Cat? Cats move to, like, we've got this empty room, kind of in the middle of the room. Cats move over to this side of the room, dogs move over to this side of the room. Let's go ahead and cluster up. You're gonna Okay, got a lot of dogs, we got some cats, equal-ish distribution. Okay, the next thing you're gonna do is you're gonna go find your cat person versus dog person. Now you're going to find somebody with an opposite opinion of you and convince them of the superiority of a cat or a dog. Okay, so just go ahead. You guys can cluster into groups. It doesn't have to be all be one-on-one. Introduce yourself first, though. Provide them your name and any additional information you like. Go ahead.
Yeah, I mean, that's.
I feel like they might say something about it. I don't know. Yeah. The cats. Trying to take online and we don't touch anything.
Okay, lots of good conversations going on, but let's go ahead and bring it on back. Find your seat when you find a natural lull in your conversation. So go ahead, find your seats. We're going to start learning about the brain, but the conversation is great. I'll honestly probably continue to do icebreaker-ish types of things for the first couple of weeks of class, for a few different reasons. One, to build community. You guys are all going to be in this like same cluster of humans, like for this semester, for next semester. Usually by the time we get to 281, everyone knows each other pretty well. And so it's just nice to have that like open dialogue. And then if you are a neuroscience major, you're going to continue to see these individuals through your 380 classes and all of those upper division electives. So it's just nice to feel like you have a set of people that you can go to if you have an issue or you're going to miss a class or something and need notes. So we're going to continue to do that. So thank you so much for participating and chatting and yeah, not making it awkward silence. Good job. Okay, so where did we leave off on Tuesday? So we started to do a little bit of anatomical review, which was where we're going to kind of jump off today. We're going to do a little bit of an in-class activity. So you will at some point in time be paired up with another individual. I'm not going to pair you up. I'll let you choose who you want to work with. And then we'll get into some of the historical kind of references of neuroscience, kind of how this field was developed across the ages. So let's go ahead and get started. Okay, so this is week one. Really quickly, just to kind of get yourself in the rhythm of this particular course, every week you are going to have a quiz. You will also have a study guide that you'll be required to fill out. The study guide I just kind of posted here in terms of things to make sure you're attending to. You'll be answering these study guide questions. We're going to discuss some of these study guide questions today in class. As I said previously, almost everything on these study guides is taken directly from the slides. So as long as you have the slides available, you should be able to go through and find what you need to find. You guys I presented the syllabus kind of day one, and maybe you all had an opportunity to look at it as well. Does anybody have any questions about kind of classroom processes, expectations, anything like that? At the end of today, we'll talk about kind of the final project for the class a little bit, but right now I just wanted to ask if anybody had any questions. No? Okay. Well, if you do, feel free to reach out. Somebody did notify me that all of our quiz dates were set to previous years and all of our assignments were set to previous years, which was probably pretty annoying if you were getting notifications. So I sat down yesterday and updated all of that. So it should be all reflective of when these due dates for these quizzes are and should all be aligned. So hopefully that's made your life a little bit easier. But if you ever do have an issue like that, where there's something that's visible on Canvas or there's a due date that's incorrect, always feel free to reach out. Never feel like you're bothering me, because if you're seeing it and identifying it, I'm sure somebody else is, and I'd rather get it fixed sooner rather than later. So thanks. Okay, so on Tuesday, we left off talking about kind of the 7 major regions of the brain. Again, this is kind of just introductory neuroanatomy. We have our frontal lobe, our parietal lobe, our occipital lobe, our temporal lobe, kind of all of like the surface features of our cerebral cortex. Last time we also talked a little bit about the differences between the cerebral cortex, aka the cerebrum, that we see up here, and here's the cerebellum, and then we have these regions of our brainstem. Now, keep in mind, as humans, we have a very developed cerebrum. We have a very developed frontal lobe. This we can see within the different species can shift depending on the amount of brain matter that they have related to those functions. But almost all mammals you're going to see have these brainstem features that are so important for mediating kind of central functions to living, right? Our brain stems are going to carry and produce or send out information to make sure that we keep respirating, that we keep breathing, that our hearts keep breathing. So if you have an injury to your brainstem, tends to be pretty detrimental, because you're going to interfere with very primary life functions. Now, if we start here kind of down at the bottom, this is a very condensed version of the spinal cord. Your sacral region of your spinal cord is obviously going to be here down at the base of your spinal cord. Then you have your lumbar region, your thoracic region, and cervical region. As we talk about things like the neuromuscular junction and motor function, we'll talk about these different cross sections of the spinal cord and kind of what they correlate to. Keep in mind, when we're looking at the spinal cord, it kind of serves 2 different functions. It's going to be sending information out to your body to tell you to move, right? So if you move your little finger, that information is coming from your brain and going through your spinal cord and projecting out to your finger. But if you're going to take that same finger and touch the desk or prick it or get a paper cut, that same information is going to be traveling up through your peripheral nervous system, up to your spinal cord, and then up to your brain. Keep in mind, different types of information will travel different pathways. So if you burn your finger on a stove versus if you cut your finger, or if you are just pressing your finger, those are all different types of information that have different types of receptors and different pathways that they travel. We'll go into that later, but it's kind of good to lay that foundation now. And then if we come up here to the basis of our brainstem, we have again our medulla oblongata, our pons, shown here, as well as our midbrain, which kind of spans between the midbrain itself and then those projections out into the cerebrum. This is our cerebellum. Our cerebellum is super important for balance. It gets direct projections from our ear. Why would it get direct projections from our ear? What? I heard someone say quiet. Oh, yeah. What was it? Is it Sophie? Yeah. So it's near the temporal lobe, absolutely. All of our auditory information goes to that temporal lobe to get processed for auditory information. But you guys have heard of like that inner ear, that cochlea. Yeah, Mitchell.
Like balance.
Balance, yes, right? Have you ever had an ear infection and just not felt quite right? Like that inner ear, we've got there's a liquid in there that helps us maintain balance. And that projection goes directly to the cerebellum and helps us maintain balance. And so if you look at a cross section, if you actually did like a mid-sagittal section of that cerebellum, it's kind of wild, because if you cut it open, it actually looks a lot like a cauliflower. That's what the structure of it itself looks like. Now, if we project further up into regions like the diencephalon, the basal ganglia is going to be super important also for motor movement and mediating that. Deficits in the basal ganglia, we definitely see with certain disease states, one of them being Parkinson's. And so that's how we all can also relate the function of a region, is when we see deficits related to specific disease states. Now, if you do kind of a cross section of the brain, you'll notice like we have a lot of cells on the very surface of the cerebrum. And then inside of that, you'll see a lot of projections from other areas. And we don't have a good cross section of that here. We might have it on some subsequent slides. But again, we're not going to get too down into the weeds of neuroanatomy. I'm just trying to give you some ideas of where these different locations are. So once we do get into structures, it's not a completely foreign concept. So yeah, so here we see another cross section of these functional regions here again, our spinal cord, our medulla oblongata. This is again kind of a mid-sagittal section, cross section that we're looking at here. Our most superior region of this is going to be our cerebral hemisphere, and our most inferior region that we're looking at on this slide is going to be our spinal cord. So just so we start to get used to using those words. We'll talk more about some of these different substructures again as we're going over specific functions of those regions. Much like when you look at anatomy or really any biology, there's a very close kind of structure function relationship. That's something that we'll talk a lot about. Okay, so what are the functions of some of these regions? So that's me kind of posing a question to you. So I'm going to just have you turn to a neighbor. You can use previous knowledge from other classes that you've taken. If you are completely stumped, totally fine. Feel free to use the Googles. I don't care. But I'm just going to leave it there. So go ahead and turn to the person sitting next to you. Choose one of these regions that we see here and go ahead and identify one of the functions of one of those regions. Okay, so go ahead and chat it up. Oh my gosh, could I? Okay, does anybody want to share out a region that they selected and the function of said region? I do know all your names. Yes.
The midbrain's control, so it controls some voluntary movements like walking and running.
And you said midbrain?
Yeah.
Yes, nice. Okay, does anybody else want to share anything out? You know?
The spinal cord is responsible for a lot of the balance and support, as well as receiving, sending out nerve signals to the brain and body.
Yeah, so all of that sensory information, especially things like proprioception or the sense of touch, is definitely going through that spinal cord. Yeah, that's great. Mitchell?
Yeah, the corpus callosum that connects to.
Yeah, the corpus callosum is literally a set of fiber tracks that are going to cross at the center portion of the brain, and it's going to be able to transmit information between those two hemispheres. So yeah, absolutely. Thanks for sharing. Okay, I'm not going to press you too much. Thanks for the folks who shared out. We're going to keep going here. For these different areas. So for the surface of the cerebral cortex, and again, make sure we're keeping this very clear. Our cerebral cortex is kind of what's shown in this image here. We're missing the brainstem as well as the cerebellum here. So that's our cerebral cortex area. We have our frontal lobe, our parietal lobe, our occipital lobe, and our temporal lobe. Each of those lobe regions, in fact, does have a function. So what are some of the functions related to these regions? We only have 4 regions here. So kind of same thing as before, just choose a region, identify a function, and then I'll have you report out again. So go ahead and discuss quickly. Choose one of the regions and yeah, just identify what its function is. Okay, so what did we come up with? For our frontal lobe. Frontal lobe would be associated with what types of functions? Your frontal?
Yeah, it's your personality, it's what makes you, you,
Mhm.
your decision making, stuff like that.
Yeah, decision making, personality, critical thought analysis. Do you have something to say?
I just like, planning and like cognitive, higher cognitive skills.
Yeah, higher cognitive skills. So as humans, we have a pretty developed frontal lobe, right? That's one of the things that's going to differ us from other mammalian species is this region of frontal cortical matter that we have dedicated to things like decision making, forward thought. We will talk about some types of memory entrainment occurring in the frontal cortex in second semester. So yeah, the frontal lobe again is you can think of kind of what some of the more higher cognitive functions that we carry as humans that maybe not other species don't have as developed, but still might exist within them. Parietal lobe. Yes.
Our spatial navigation, motor control, attention, perception, sensory integration, lots of language and communication.
Yeah, that's a lot. Yeah, absolutely. Language and communication is something we'll talk about a little bit later today. And I just, both the somatosensory and motor cortex actually exist right along this region that we see right here. And so those are going to be some of those primary functions of the parietal lobe, but you gave a good list. Good job. Abby, right?
Yes.
Okay. Anybody want to share out occipital lobe? I feel like is a good one because it's pretty easy. Vision. Thanks, Ben. Yeah, so our occipital lobe is going to be important for visual processing. So we'll talk about, it's kind of interesting to think about how the sensory system that comes in through the front of our heads essentially, or that, visual part, hits all the way back onto the occipital, gets projected all the way back into the occipital lobe. But it does have a couple stops and gets processed in several different regions just because it's so important to us, evolutionarily speaking. Temporal lobe. Let's stop playing with that. Yeah, hearing, language, things like that. Thank you, Robert. So in our temporal lobe, we've got auditory processing that occurs there. We also have a region of the brain called the hippocampus, which is super important for memory entrainment. And then also our sense of olfaction or sense of smell also gets processed in that temporal lobe. And so there's actually a really tight connection with memories and smells, just kind of due to the location of both that area of the brain called the hippocampus, as well as that kind of olfactory processing. Again, we'll talk about olfaction and all of our special senses in the second semester. So yeah, if you were kind of following along and trying to take notes, here we also have kind of just a short synopsis of each one of those regions. Again, frontal lobe, short-term memory and future planning. Personality is also a component of that. Parietal lobe, somatic sensation. We do see language, production, processing also occurring in that kind of parietal frontal lobe region in the left hemisphere. And then the temporal lobe, auditory processing, deep structures for memory and emotion. So we have both the hippocampus, which is important for memory, and then another region of the brain called the amygdala, which is going to be tied to a lot of emotion and emotion regulation. And like I said before, occipital lobe, pretty straightforward for the most part, just primary visual processing, and then some secondary visual processing might occur there as well. Okay, so again, as we're talking about neuroanatomy, it's good to make sure we feel comfortable with all of these terms. Sulci and gyri. Sulcus is like the valleys that you see in the brain, because if you look at the surface of our brain, obviously it's got those bumps on it. So the valleys are going to be your sulci, and then the peaks or the tops of those surfaces are going to be the gyri. You can see here that there are a certain number of kind of larger kind of landmarks of the brain that make it easy for us to divide it into these different regions. So for example, the central sulcus is very clearly apparent on human brains. It kind of divides the frontal lobe from the parietal lobe. And then on each side of that, we have our motor cortex that's kind of in front of that central sulcus. And then we have our somatosensory processing, which is kind of more a part of the parietal lobe. That is, again, considered the central sulcus, the valley that divides the frontal and parietal lobes. The precentral gyrus is going to be kind of the green region that you see highlighted here. That's important for the motor cortex. And then our postcentral gyrus is going to have our somatosensory information that's processing there. The other kind of large landmark that you should see on the surface of a human brain would be this lateral sulcus that divides that frontal lobe and temporal lobe. Again, it's kind of just like these very clear landmarks. If you're looking at a human brain, you can really kind of see the differences in these different regions that we're discussing. So again, this isn't a neuroanatomy class. We're not going to get in and to learn every single sulcus and gyrus and Brodmann's numbers that correlate to them. That's just not what we're doing here. But it's good to know kind of these bigger landmarks. So when you do get into a neuroanatomy class, you can be like, ah, yes, I do remember this. Yeah. Okay, in terms of hemispheres, we have our left hemisphere and our right hemisphere. Controls kind of the contralateral side for motor movement and even sensory processing. When I say contralateral, again, this is a term that we use in anatomy, and if you haven't taken an anatomy class, contralateral basically means opposite, right? And I think if you think about this, we probably know that, you know, the left part of your body is controlled by your right hemisphere, and the left hemisphere controls the right side of your body. So we have this kind of move, what we call decussation, or crossing over of these pathways as they project up into the brain. So that's that when we say contralateral, we're talking about the opposite side. If it stayed on the same side, the term that we would use there is ipsilateral. Okay, so contra is opposite, ipsilateral is same. And again, we won't harp on those terms too much right now, but we will come back to revisit them. So sometimes it's just nice to have them be familiar to us. Each hemisphere is not completely symmetrical nor equivalent in function. You do see some, we'll say, hemispheric differences. So let's say, again, we see the contralateral control of the sensory of the motor systems from the opposite side of the body, right? Also tends to have a basis in creativity, spatial ability, context and perception, and then recognition of faces, places, and objects. So if you're not great with faces, places, and objects, maybe there's something going on with the right hemisphere. Left hemisphere, we'll talk about later today, speech, language, and comprehension. Those are actually kind of two different regions of your brain. We're going to talk about one region that's important for comprehension of speech, so how you know what I'm saying. And second would be speech production, the area of our brain that's important for you to be able to produce speech as a response. Analysis and calculations, timing and sequencing, and again, recognition of words, letters, and numbers are a little bit more kind of that left hemisphere. And from these hemispheric differences, that's where that idea of kind of right brain versus left brain kind of comes about. Not entirely true. Keep in mind, one thing that we've learned about the brain is that there's a tremendous amount of plasticity, meaning that if you lose function of a specific region of your brain, there's possibly an opportunity for another part of your brain to kind of pick up that functionality. Ooh, okay. So the next thing that we're going to do is our in-class activity. Before we do that, I just, again, going to make you talk to somebody different this time. This might include some twisting. What do you think? Is your left or right hemisphere higher functioning for you? Or which one do you feel like you personally kind of attuned to more? Are you a little bit more, okay, right brain versus left brain, but those again, terms that probably don't exactly apply. But which hemisphere do you feel like kind of vibes with you a little bit more? So try to talk to somebody different. You can go back, I'll let you go back to your kind of comfort group here in a second, but try to find somebody different maybe to talk to. So if you need to stand up, totally fine. So just take a few seconds and discuss. Mass.
I firmly feel like
It's hard to.
Bring in and Annabelle.
I've absolutely, but I'm also like,
I
subjects have always been like English and mine, which don't really make sense together. And I don't know, I've always just said that's because I am in doctors. I probably have not had yours anything. Yeah, that's what.
Okay. Okay, so, great, you got an opportunity to chat with somebody else. Okay, so the next thing that we're gonna do is I'm gonna break you out into a little in-class activity. The completion of this activity will earn you 10 points. That'll be your first kind of 10 points for the semester. And so let me just give you some clear directions, hopefully to help you out here. This activity will require 2 devices, okay? So if you want to make sure you assemble into a group that has at least 2 laptops, okay? So if you need to make a group of 3 or a group of 4, that's completely fine. So this in-class assignment, if you're following along in the slides, there's a link here to complete this in-class assignment. If you go to our Canvas page, there is also a link to this website. And I'm just going to quickly hop out here and kind of show you where this sends us to. So this is going to send you to this amazing kind of anatomy website that the University of British Columbia has put together. It does a really nice job of showing you all of the different structures that we have covered. I'm going to give you some class time to kind of work your way through this, basically kind of orienting yourself. What's nice is it also has these kind of opportunities to interact with the website itself. This is just really nice for learning this foundational neuroanatomical information. So the first thing that you'll do here is find a partner. You'll visit our Canvas page for the link, or you can click on the link that's in the PowerPoint. You're going to work your way through this module, so you can kind of see that again, there's all of these different module points, and then you get down here to the very bottom, it'll ask you to test yourself. And so once you get to test yourself, it's going to ask you a series of questions about these different regions. So what is region 1? Okay, so what I've done is you get to test yourself on the website. But if you go to our Canvas page, there is also kind of like a test yourself quiz. So that's why you need kind of the 2 different devices, is because one person is going to be going through the test yourself on the website, and the other person will be reporting out those test yourself answers on the Canvas page, okay? There's a little discrepancy, I think, with the CNS2 regions just because of the drop-down menus, and it wasn't perfectly translated into Canvas. So just work through it until you have a question, and then when you have a question, just feel free to kind of raise your hand. Once you finished kind of that test yourself, complete it on your Canvas page, and then maybe also have your partner complete it on their Canvas page as well. Okay, so yes, it's going to take a little bit of tech lifting here. But before I kind of let you kind of start to try to work yourself through this process, does anybody have any questions? No. Okay. We're just gonna, I think this is gonna be one of those experiences where we're just gonna move forward. And as you have questions, raise your hand and I'll come to you and we'll work through this. Yeah, I'll give you probably 20 minutes to do this. We'll see kind of how, as you complete it. So yes, just raise your hand if you have questions and go ahead and get started. If you have 2 people, that's fine. If you have 3 people, that's fine. Yes. The in-class activity says you need an access code. Well, that's fun. Not really. Okay, let's figure out what that is all about. This part or this part? This one? Oh, Okay, well, let me just get rid of that right now, so you have no issues. Access code, access code. Boop. Okay, if you have your kind of modules listed there, go ahead and refresh and now click into it. So you should no longer require an access code.
Yeah.
So we're supposed to experience like a couple of things. And then we all have to do these. Like, so many sort of instances. Okay. You wanna do the first one?
Yeah.
You know what the possibilities is like EVs are all like. Yeah, we're on there. Soon that I think that's. So I think rise will come out also. Right. Yeah. There's no sucker really. It's right. They're together. We got like central focus. Gens, I guess. Yeah. So yeah, those are the regions of it. Lateral fissures and reefs. Okay, just see. Because I see. Yeah, this is one word. And it's yours, I. Margins are fissures. Yeah. Separates the weight. Yes, yeah, exactly. Basically, it's like, yes, or separates, yeah, right there. So that's. Function. Name and household. Oh, use that option. Yes. So, if there are any questions, you guys are like, we're just gonna work through this, we're just gonna answer the questions, which is totally fine. If you get to question 3, which is where this is gonna get hard, tricky. The wording there's one option that's correct and one option that's clearly incorrect. So just choose the words that kind of make sense with the structures that we have discussed, okay? Because I just couldn't get it to, because having the ordering of those words, there's a lot of varieties of combination that could have gone. So just choose the correct word for question 3. So just.
We can do it.
Okay,
You want it, okay.
you work through the whole
Okay.
thing and then answer. But we've been doing like back and forth, like you go to the question, look at.
I'm gonna try to highlight the areas and drop down the names.
Does that make sense to you? And.
Or is it just this one after, yes?
No, it's 10-12. And then it's on line, sort of, or front one dial, which is one.
I don't know. What was this one? I think that's the central list. I'll check this one.
Oh, you. Yeah, I know.
Yeah. That would have been great. Yeah.
Is that one word we thought supposed to be on?
Yeah.
Brain and your medulla.
Yeah, actually great.
Okay.
Yes. Okay, so now we got.
Yeah. We're fine.
Yeah. So one.
This is. Do we look at the, what were the test results? This kidding. So it's like. Out in the way.
So central. Yeah. Yeah. The lateral. This one. So separates the. Or wait, it'd be frontal and.
I'm seeing the, seeing the vision. I'm seeing visions.
I'll say families through knuckle bones.
Horrific.
So if they don't. So. Right on the left side. Okay, on the left side.
So like, we split up. Maybe this. Oh, maybe. Not.
Central separates the fallen both spreadal and the, oh, yeah. Yeah. Separates the varietal and occipital, right? That was the, or no, wait, that was this sideways one, sorry. Separates
The arrow is like not. But just not. Like what? Good. Where's that? It's correct. It's correct.
Yes.
This is. And I. Comparative from the room of today's design. Ten is. Yes.
I guess it'd be varietal. And then the.
Okay, so big, and then it's not bigger. The four is not. So seven is. For. Like promise or. I think she's just. It's right here. It's right.
Are you getting-
Get it to stay.
He opted for this, but that was.
Yeah.
Yeah.
So you can't get a stick.
Yeah.
Is it like you get to click on the thing?
We'll just do it by process elimination. Boop.
Maybe you have to start from one.
Oh.
Yeah. Okay, this section here, CS4, skip it. Just skip it. Okay. Just skip it. It's just gonna frustrate you, and I'll be honest, many of these substructures, we will not be going into a ton of density about. So this particular one, skip. The rest of them are a little bit easier. We can work through pretty quickly. This matching will probably drive you a little nuts. But if it's, again, if you are super interested in neuroanatomy, your future is as a neurosurgeon, by all means, you could spend all day here if you like, but I would just skip it for the purposes of today's exercise.
Wait, I'm sorry. Because that's also what.
The plane is just the yellow thing. That's what it's asking about. That plane.
Thank you, because we were looking at it, we were like, there are multiple brain deaths that happened here.
No, for sure.
I was like, we're gonna get you one thing.
Seriously though. This should be the.
Oh, it's especially because central is very.
Yeah.
Oh, thanks.
Yes.
And now I didn't see that.
I know, I was just making sure.
Okay.
Here.
there isn't one, at least one part that's incorrectly keyed on this in-class activity, so do not take whatever score you get on this too seriously. You are going to get full credit as long as you participate. And that's really the way it goes with the in-class activities. As long as you show up and participate, you get your points, okay? So whatever the score comes up with after you've selected all your answers, I'm just gonna go in and rewrite those all to 10s, okay? Not to say that you shouldn't take this seriously or learn the material, I'm just letting you know what's gonna happen.
Yeah.
That probably goes back to the that. Because I was so scared.
We did it.
Or it's just like.
It's different.
For saving.
There's one of those. That one's wrong, but put in the right answer. The partial.
It's making me have too much time.
Okay.
So really quickly, I would like to go back into lecture mode now, and I know maybe you haven't had time to shift and have the other person of your group maybe complete all those answers. This in-class activity will stay open for you to complete on your own time. So you can do it later today, you can do it next week, I do not care, just make sure you get that in-class activity done. And then I will update your grades all to 10s. Yay. Really quickly, this website that you guys were just playing on, this is, as I said before, it's actually a medical school, I think, the neuroanatomy course for medical school in British Columbia. This is kind of the homepage for it. If you have interest in neuroanatomy, great website. They've got multiple things here that are available to you in terms of interactive modules, a stroke model, things like that. So if this is your jam, come use this website. It's kind of phenomenal. Okay. Everybody okay if I continue to move forward? Cool. Okay, let's go. Okay, so that's our first in-class activity. The purpose of an in-class activity is to give you, the students, an opportunity to kind of dig into a topic a little bit more, maybe works alongside one of your classmates, allow you an opportunity to process this information and maybe self-assess. So we'll be doing a few more of those across the semester. Okay, so now we're going to go back into straight into lecture mode, and we're going to talk about the history of cellular neuroscience and kind of how did we get to where we are now in terms of our beliefs about the brain. Okay, so if we go all the way back to the 2nd century, Galen proposed nerves convey fluid secreted by the brain. It was just, basically brain and fluid. If we go to the 1800s, this is again kind of the origin of cellular neuroscience. Golgi, who invented the Golgi stain, actually developed a staining technique that allowed us to identify individual neural structures. So here we have Ramon Cajal, who put this into application. And these are pictures that he drew of the stain that he did in the 1800s. These are live images of neurons that have been taken in the last, 20 years, right? Not a huge discrepancy there. You're actually seeing a really nice snapshot of what the cells in the brain look like, okay? Now, this is more of an aside than anything. One of the beautiful things about the Golgi stain is it doesn't stain every single neuron. It selectively stains, just a few neurons, which if you're looking at a brain slice is actually incredibly advantageous because if it stained every single neuron, it would just all look like one big black smear, okay? So having that selectivity is super important, and only recently have we become really good at being able to look at an entire brain structure with every individual neuron having a different color. And that was basically accomplished using a set of expression systems called the rainbow. But this started to give rise to the neuron doctrine, which states that individual neurons are kind of the building blocks of the nervous system, that these individual cells are important for neuronal function. In the 1920s, Harrison axons and dendrites are basically looking at these extensions from the cell bodies themselves. That's what those axons and dendrites are. And then in the mid-1950s, Sandy Pele used electron microscopy to look at individual synapses or connections of these neurons. So this is, again, more of a kind of visual walkthrough. How do we know neurons are the cells of the nervous system? So again, we know that there are fluids. Then we invent a staining technique by Golgi and Cajal that allowed us to look at individual neuronal structures. And then you were able to look at axons and dendrite projections in the 20s. And then in the 1950s, we were able to actually look at individual synapses. So we're just kind of zooming on in as to what's happening with these cells. Now, if we go, oh, this is just distracting. I'm getting rid of it. So here we're able to look at kind of the history of the mind, body, brain philosophy, right? This is where neuroscience kind of starts to, we'll say, go into other areas of study. So first we have Descartes, you have your dualistic theory in the 17th century. Your brain mediates perception, motor acts, memory, appetites, and passions. But the mind is really what's controlling kind of your higher cognitive functions and is controlled by your soul. If we move forward to the 18th century with a series of empiricists kind of led by Immanuel Kant, this is where that concept of us coming into this world as a blank slate, aka tabula rasa. If you've taken a philosophy class, you have probably heard that term before. So we come into this world and, a blank slate, and then we have all of these sensory experiences that help us kind of become who we are going to become. Keep in mind, a lot of these beliefs are historical. We've since disproven some of those ideas, so it's kind of interesting to think about what has persisted and what has evolved or changed over time. Believe that our perception of the world is determined by our inherent features of our mind or brain, and we'll also start to talk about kind of phrenology as a concept of neuroscience, and this kind of structure-function relationship, which I've already alluded to is important in neuroscience, but phrenology was a little off the mark. We'll talk about why. Lastly, in the 19th century, Darwin came on the scene, also writing about not just the process of evolution, but also the brain as the basis of behavior. And then within the 20th century, we saw Freud come about, psychoanalysts. We've discussed the id, the ego, and the superego, and their relationship to who we are. Yeah, your psychology professors are kind of cringing as to how I just kind of, went over Freud pretty quickly. Okay. So anyway, if we go into kind of another viewpoint at which the brain has been analyzed, we first talked about kind of cellular neuroscience, then we had philosophical neuroscience or kind of philosophy of the mind, and now we're going to go into kind of the electrophysical kind of perspective of understanding the brain. And so people who study the electrical currents generated by the brain are called electrophysiologists. And we'll talk more about kind of the electrochemical gradient and how our brains actually generate these electrical currents later on. But this was first observed in the 1700s by Luigi Galvani, where he was able to observe nerve cells produce electricity. And then if we fast forward to these 19th century Germans, they were the first individuals to kind of measure the speed of conduction along the nerve cell. Keep in mind, one of the first characterizations of understanding the way that neurotransmitters even work was our ability to isolate acetylcholine and apply it to muscle cells, as you can see kind of here with the frog. And that was one of the ways we were able to identify the role and responsibilities of acetylcholine. Hodgkin and Huxley, back in, we'll say, the 1950s, the first researchers to identify the action potential and all of the ion channels responsible for the action potential. Fun fact about me, I am such a nerd that my cat's name is Huxley, after Hodgkin and Huxley. And then I got a second cat, and I should have named it Hodgkin. Did not. His name's Steve. Anyway. And then most recently, we have Benderlin LeBay, who is through surgical interventions, has started to actually be able to observe cognitive processing and the relationship to electrical processing and kind of the timeline that appears there. So here we can see an EEG, kind of the readiness potential onset is like at 0.5 seconds. I decide to lift the finger is kind of cognitively processed around 0.2 seconds. And then at 0 seconds, you actually have that finger lifting. So being able to kind of come up with a timeline between cognitive intention and when the actual action takes place is something that we've just more recently been able to really measure. Questions? Okay. Yeah, Debbie?
How do you measure, like,
People-
how alcohol and drugs affect that? Like the actual timeline, I'm sure you know, like, is it a stark difference? Like, a whole lot longer, or?
Yes, you can look at something like that. So for example, you just threw out alcohol. You could study the effects of alcohol at several different levels, right? Sorry, I'm gonna get into the weeds here for a second, so just listen and we'll reel it back in. So when I say at different levels, you could study how ethanol binds to the individual receptor and how it affects the opening and closing channel properties of that individual receptor. That receptor is going to be expressed on the surface of an individual neuron. You could actually measure action potential depression of that neuron based on the alcohol that has bound to that receptor, right? And so you can go individual protein, you can go cellular function and the impact it has on that projection. And then as we were just looking at down here, like this is higher cognitive processing and reaction times. So at the human level, you could obviously do almost like a dose response curve of like, as we increase the amount of ethanol that this person consumes, how does that affect their reaction time for specific measures, right? So there's like several different ways and methods that you could analyze that. And I'm guessing if you did it at those different levels, you would probably see it be pretty similar, right? You could say, oh, it's affecting the, or it's opening up GABA receptors at a higher rate. And so that's going to increase or decrease the potential of those axons sending action potentials, thus also slowing the reaction time at like the muscular level. So yeah. Sorry, like I said, I'll do that whenever you have a question like that in this class. That's why we're here, is to have the fun kind of offside question. So please, thank you for asking that question. And maybe it'll stimulate something in your mind that you haven't thought about neuroscience too. So yeah. Okay. So I talked about phrenology on a few slides ago, and this is where we're coming back to revisit it. And phrenology was originally introduced by Franz Gall, a Viennese physician and neuroanatomist. So he really had kind of like two different ideas. One of them being good, this idea that the brain is the organ of the mind and all mental functions emanate from the brain, not kind of that mind-body dualism that we mentioned a moment ago. So that was good. He argued that the cerebral cortex did not function as a single organ, but contained within it many organs, and that specific particular regions of the cerebral cortex control specific functions. And behind that, I kind of put meh, because it's like kind of true, because it's starting to kind of touch on the whole structure-function relationship. But the way that it manifested in his proposal was that you have kind of this like legit idea that you have this substructure function relationship absolutely on the money there. But the bumps on the skull, that's where it gets a little yikes. Because in phrenology, these individuals would literally like put their hands on the surface of your skull, feel those bumps, and be able to tell you, oh, this is where your ideality exists, and you've got a bump there, so this is how it manifests in you. So they were looking at the surface of the skull and not the underlying cortical matter. Okay, so that's how phrenology is kind of like, but the good thing of phrenology that came out of phrenology is, again, this idea that you have maybe these subregions of the cerebral cortex that have different functions. So phrenology, a discipline concerned with determining personality and character based on the detailed shape of the skull. And you've probably seen like these phrenology kind of dummy heads, because they're pretty popular to like decorate with. Oh, I also started throwing pictures of these people up here because you always learn history, but you never see the people who've done it. So this guy looks like a lot of fun. I'm like, you're like, yeah, so severe looking. Great time at parties. Okay. Oh, Pierre Florens. Okay, so he took Gall's idea a step further about this kind of structure-function relationship, but he started to test on animals this idea. Determine specific regions are not required for specific functions. That's me again, my attempt at humor. The sigh, like, ugh, no, that's not exactly what's going on. That's again kind of taking a step back from phrenology in terms of this structure function relationship, and that he proposed that any part of the cerebral layer can complete any function. Okay, meaning that your visual control could occur in your frontal lobe, which now we know is not true, right? You have very specific substructures in your brain that have very specific functions. So it's, not surprisingly in history, you see this often where you'll take a step forward in a field, and then maybe sometimes a popular opinion will come about that you'll take a minor step back. So these findings and cultural influence led to kind of this holistic view. They rejected the human mind as a biological organ, that there is no soul, that all mental processes can be reduced to activity within the brain, and that the mind can be improved by exercising it. Keep in mind at the time, religion was pretty rampant and influencing sciences. And so that's, I think, where some of this rejection of some of these earlier concepts may have come from. Okay, so let's jump forward to kind of closer to where we are now. So the functionalists, this is probably where we see kind of the beginnings of kind of modern neuroscience and modern neuropsychology really taking root. In the mid-19th century, you have Paul Pierre Broca, where that might sound pseudo-familiar to you. Carl Wernicke, again, you probably know that name, some relationship. If you don't, no big deal, we'll get there. And then Hewlett Jackson's, and I like to put who because no one's ever really heard of him, but he still had a considerable impact in the field that you have different motor and sensory functions that can be traced to specific parts of your cerebral cortex. Broca and Wernicke, they have regions of the brain named after them for Broca and Wernicke's area. And we're going to talk about the function of those areas now. Okay, so this is the first evidence of a structure function localization that came from essentially language disorders. So these individuals were studying essentially speech, language, and pathology, which again, if you learn about this and this is totally your jam, we have departments on this campus that just study speech, language, and pathology. So Broca originally did this research, looked at post-mortem lesions, meaning that individuals who have passed away with these, with not just bumps on the head, but looking at what the deficits of the brain revealed if they did have these language disorders characterized as aphasias. So an aphasia is, by definition, the loss of ability to understand or express speech. Keep in mind, those are two different things. Being able to understand and being able to express are separate areas that we'll talk about here. But aphasias is kind of like the broad term that we use to describe them. And this is often caused by brain damage. It can, we'll see aphasias often will occur in times like stroke, right? You have sometimes speech deficits associated with stroke. And so that's what leads to the presentations of aphasias. And so this is an image of Pierre Broca that you see here. Okay, so here's a quick case study. No, if you do a case study today. A patient has a stroke, is unable to speak, can understand language perfectly, has no physical deficits that would prevent speaking, can sing, whistle, utter individual words, and Broca studied his name post-mortem. Where was, well, this is her, but where was their brain affected? Turn to somebody sitting next to you, come up with a hypothesis. What region of their brain do you believe was affected? Okay, so can understand language, cannot speak. What type of aphasia do they have? Or what type of the area of the brain was affected? Broca's area. So the kind of giveaway here is Broca studied their brain postmortem. But Broca's area, this is the area that is affected or does speech production. Okay. So here we have our Broca's area shown here on our left hemisphere of our brain. Okay. You're going to probably notice a pattern here really quickly. Another case study. Patient had a stroke, is able to speak, cannot understand language, has no physical deficits that would prevent from speaking, and Wernicke studied their brain post-mortem. Where was his brain affected? Or their brain? You guys are like, we don't even have to talk about this. Yeah, it's gonna be Wernicke's area. So Wernicke's area, this is the area that affects language comprehension. Okay, so Broca for speech production, Wernicke for language comprehension. Okay, those two things could get inverted quite quickly. So turn to the person that you're working with or chatting with or what have you today. See if you can come up with a fun way to remember that. Okay, because I do feel like it's sometimes easy to conflate these. Okay, so Broca for speech production, Wernicke for comprehension. So
Like, really, just like, and then there's like, somebody and do why they don't. And that they. And I was just say
okay. So hopefully you were able to kind of identify a nice way to remember those two different functions. Okay, so Wernicke's area, again, for speech comprehension, right? Surrounded by the auditory cortex and also known as an association cortex. You also is integrating information from auditory, visual, and somatic sensations as well. Keep in mind with their original model, there were kind of these initial steps for processing that occurred in separate sensory areas. And then you have these other specialized areas that also provide information. Now, between Wernicke's and Broca's area, there's actually this projection of axons. It's called the arcuate fasciculus that you see right here. You can kind of see this projection from one to the other. And that also can lead to deficits as well. So this spoken words are transformed into neural sensory code shared by both speech and writing, and also related to Broca's areas, which contains rules or grammar for transforming sensory representation into motor representation that can be realized as spoken or written language. So this theory, the Wernicke-Gershwin model, again, you've got language involved in these separate regions for both sensory and motor regions. Again, your motor regions are more for that speech production of Broca's area that govern mouth, vocal cords, throat, tongue, palate. And then sensory regions, again, is that language comprehension. Now, why are we talking about this again? Let's just like step back. Keep in mind, we've been going over kind of the historical perspective of neuroscience. And this was really kind of the first foundational research that was done to understand the structure-function relationship of something that was very easy to kind of measure or assess post-mortem in terms of deficits and the areas that were affected. But we are kind of getting into kind of like more of the clinical stuff right now. Okay, there is a third type of aphasia. This person can understand words that they read or hear, can produce words with no motor difficulties, do not correctly use the words, cannot speak coherently, and are painfully aware of their mistakes. So where was their brain affected? There's a couple different options here, but I'm going to see if you can come up with a hypothesis. Okay. Does anybody want to throw out their hypothesis? It's okay if you don't. Okay. So in this case, the type of aphasia is called conduction aphasia, and it's that connecting projection between the two. So that arcuate fasciculus or that angular gyrus gets disrupted. So you have the ability to comprehend, you have the ability to produce, but they're not talking to each other. So this individual, again, can make sounds, can make words, can understand what you're saying, but cannot communicate effectively because of that. So again, this is again the seminal work done by Broca and Wernicke. This inspired Brodmann to study cell types. And so Brodmann went through and looked at the entire surface of the cerebral cortex and started mapping out different cell types. He identified 52 different kind of structural areas of the cerebral cortex. Again, what's shocking about this is Brodmann did this characterization at least 100 years ago. We still use Brodmann's areas characterization to this day. If you go to a neurosurgeon and they do surgery on the surface of your cerebral cortex, they're using this numerical system that you see kind of represented here. So it's kind of wild. Yeah. So here today, again, the way we're able to more non-invasively look at the structure function relationship is through doing non-invasive studies using things like PET imaging, positron emission tomography, which with PET imaging, you have to have a radio labeled tag that's specific for a receptor in the brain that will only go to one region. So that is great if you're interested in, like, let's say, localizing receptors. fMRI is great because that helps you identify which regions of the brain are active. So there's a kind of different questions. And so that's why we have those two different types of methods. Cool bilingual study that was done. I mentioned this because a couple of you mentioned that you were bilingual. This work was done originally by Joy Kirsch. This study was done to identify during language development, right? We've got this critical period in which we all learn language. Usually that begins around from birth all the way up to, we'll say, like, I don't know, the lower ages of like 7, 8, 9. You just have this period where your brain is very open to acquiring a second language. So on the left, we have an individual who was an early bilingual. They grew up in a household that spoke both Turkish and English. And what's interesting here is if we're looking at these bilingual speakers in this subregion of their brain, the two areas in which they learn those languages completely overlaps. Now, if we look at a late bilingual, what's interesting here is this person learned French secondarily later in their life. Their native first language was English. And what we see here are completely discrete areas of the brain that are being used for this language. So it's really interesting in terms of like critical periods and neuroplasticity and how our brain changes over our lifetime and kind of how that gets represented into these areas of the brain. I will tell you, I've taught this class, I don't know, for like 5 years. This study is the study that all my students talk about like years afterwards. They're like, I remember the cool bilingual study. That and the three-eyed frog we'll talk about next semester. We'll get there. Okay. What's interesting about this is it's actually translated to different types of language as well. So for example, American Sign Language uses hand gestures to convey letters and sounds rather than sight and sound, but has the same structural complexity as spoken language. What's interesting is just like spoken language, this is localized to the left hemisphere. Deaf people can become aphasic for sign language, often as a result of lesions of the left hemisphere. So the types of aphasia that we just talked about for spoken language also occur in individuals who speak ASL or use ASL. And damage to the left hemisphere can have quite specific consequences for signing, just as spoken language. Sign comprehension can occur if you have damage to Wernicke's area. And then again, grammar issues can also occur if you have damage to Broca's area. So again, these aphasias that present in spoken language also translate to ASL. And I mentioned, I think on Tuesday a little bit, that babies who learn ASL also babble as well. That kind of development of those speech patterns occur in the same way. Okay. Another thought about brain processing, again, very big picture. This kind of wraps up our historical perspective, is as we talk about how the brain functions and communicates, if we kind of shift this over to almost like computer/physics talk, our neural pathways have what we call serial processing. Our sensory systems are a great example of that. I kind of ran you through again the very quick visual, projections of rods and cones, X, Y, and Z, right? You go from step A to step B to step C, or neuron A to neuron B to neuron C. That's a very clear linear path, right? But we don't just process sight. That's not the only thing you're doing right now. You have all of those other sensory systems that are also communicating with you at the same time. So that's considered parallel processing because you've got all of your different systems
sending information kind of in these parallel super highways. But keep in mind, you can kind of step out of that and identify individual signaling processes and pick apart the kind of serial communication. So the brain does both of these things at once. It's quite amazing in that way. And so we've got both serial and parallel processing centers, and damage to one area doesn't stop kind of the whole thing from working. Unless you do some damage to the brainstem, which might actually stop the whole thing from working. But outside of that, really you have these abilities to kind of compensate. Okay, how much time do we have left? Great, we've got a minute. So think about your grandmother really quickly. Hopefully this is, or somebody important in your life. The reason I'm telling you to think about your grandmother is because everything you know about your grandmother, for example, seems to be stored in kind of one complete representation, right? That is kind of equally accessible whether you see her in person, hear her voice, or simply kind of think about her. Our experience, however, is not kind of this faithful guide to how knowledge is stored in memory. We'll talk about how our memory can play tricks on us. But it's not a single representation, but you've got all of these different areas of your brain that are kind of activated, right? The smell of your grandmother, the sight of your grandmother, those are all separate regions, but all come together kind of into this cohesive concept of her. So, think about her ability to recognize her voice is mediated by yet another region. But this is only the beginning. The tools to answer our questions have just recently become available. We just need to ask the right questions. It's a super fun time to be part of neuroscience. And we might maybe even address the white whale of consciousness of neuroscience at some point in time, which is consciousness. Okay, last thing, I know it's 3:20, so I'm just going to say this very quickly. Our final project, you're going to develop an in-class activity that should be interactive in nature. You should try to work together with a group of 4 to 5 students. Those groups can be smaller. You're going to select a topic that you found interesting across the semester and develop this 10 to 15 minute presentation. There's both the information about the directions to the in-class activity, your grading rubric, as well as a sign-up sheet. So if there's a topic you're like, I really want to do active potential stuff, and I know that today, you can sign up. We'll come back to this throughout the semester and give you more information, but I'm gone over time. So thank you for your time today. I will see you next Tuesday.
Yeah, Michael's. You're on a long standing. I know, yeah, me too. I'm terrible at sewing because I bored.
Thanks, Josephine. Thanks for zooming in. I'm sorry I wasn't able to answer your questions. I was kind of in the mode of it all. But I hope I get to see you next week. But if you have any questions as you look over the syllabus, just let me know, okay? Okay, cool. Have a wonderful day and feel better, okay?